My name is Savannah Grogan, M number M00224988. Circulatory system, heart structure, and function. Functions of the circulatory system are to transport, so the delivery of nutrients and oxygen to the tissues and the removal of metabolic waste and carbon dioxide, regulation, distribution of hormones and temperature regulation, and protection, clotting after injury, and immune response. So transport, the blood is responsible for going into the capillary beds of the tissues of the body and taking the waste out and delivering the oxygen to them. And then the blood that is carrying the waste that's deoxygenated goes back up and gets rid of the bad stuff and then goes to the lungs to get oxygen and then take it to the body again. Regulation is the distribution of hormones and temperature regulation, and the body's temperature has to be 37 degrees Celsius or 98.6 degrees Fahrenheit, and that's important because proteins and enzymes have to be at that temperature or they denature. Protection, the blood is also responsible for clotting after injury, so it sends proteins like fibrinogen to clot. And immune response, which it contains antigens and antibodies that it then sends to places through the blood to fight the infection. The cellular structure of the heart, you have two different types of cells. The myocardial cells have striated fibers that tend to branch. They have one nucleus per cell, and they have gap junctions or intercalated discs. And once stimulation is applied, it travels from cell to cell. And then the other type are specialized cardiac cells, or non-contractile muscle fibers. These are pacemaker cells like the SA and AV nodes, and conductive cells, which are the Purkinje fibers in the bundle of His. So the two different types, the myocardial cells are the cells in the muscle that are actually responsible for the physical contraction because they have the contractile units. And then the specialized cardiac cells they don't actually physically contract. They're responsible for beginning and transmitting the electricity to the myocardial cells so that they can then contract. And so it starts in the SA node and then travels through the AV node, the Purkinje fibers, and then down the bundle of His, and then finally it reaches the myocardial cells and they contract. Heart structure. There are four chambers, a right and left atria and right and left ventricles, four valves. So you have atrioventricular valves, which is the tricuspid valve between the right atrium and the right ventricle, and the bicuspid valve between the left atrium and left ventricle. And then you have semilunar valves, which is the pulmonary semilunar valve between the right ventricle and the pulmonary artery, and the aortic semilunar valve between the left ventricle and the aorta. So basically the flow of blood through this, the blood would come in through the vena cavas and end up in the right atrium, and then it would travel down the tricuspid valve and into the right ventricle, and then through the pulmonary artery into the lungs to get oxygenated, and then it would come back into the left atrium and then flow down the mitral or the bicuspid valve into the left ventricle, and then finally it would go out of the aortic semilunar valve and go to the rest of the body. Heart structure, you have papillary muscles, chordae tendineae, and fibrous skeleton, and then the myocardial units. So here on this picture of the heart, you can see the cardiac muscle down here. You can see that it's thicker on the left side around the left ventricle because the muscle over there needs to be stronger because it's pumping blood out to the entire body. The circulatory pathway, you have two circulations. You have pulmonary from the heart to the lungs back to the heart, and then systemic from the heart to the tissues and back to the heart. And then you have vessels, and so the arteries carry blood away from the heart and the veins carry it back towards the heart. The pulmonary circuit goes heart, lungs, heart, and so that would start in the right ventricle, and then, like I said, it goes out through the pulmonary artery and to the lungs, where it gets oxygenated and trades out CO2, and then it comes back through the pulmonary veins, because it says veins carry blood towards the heart, and then it ends up in the left atrium down through the mitral valve, and then ends up in the left ventricle. And so that would be about where the systemic circuit starts. And so the left ventricle contracts, and that sends blood out through the aortic semilunar valve, and then it goes down the aorta and out through all the arteries and eventually to the capillary beds, where nutrients and oxygen are exchanged, and then it'll come back up and eventually make it to the vena cavas, where it enters the right atrium again. SA node and pacemaker potentials. Hyperpolarization is activated cyclic nucleotide gated channels family, or the HCN channel. At negative 40 millivolts, voltage gated calcium channels open, triggering action potential and contraction, and repolarization occurs with the opening of voltage gated potassium channels. And then over here, pace of the heart is set by the SA node. Depolarization begins at the SA node, travels across the atria to the AV node, travels down the bundle of His to the Purkinje fibers, and depolarizes the ventricle muscle. So here on this graph, it shows the polarization and the depolarization and repolarization of an SA node, but you can't really see it, so I have it drawn on this whiteboard here. And so right here at this line, this is where the cell is at resting potential, and it is completely polarized at negative 58. And so when the electrical impulse hits it, the HCN channels will open, and it will slowly start to leak positive inside the cell, and so that would be slow depolarization. And then once it gets to about negative 40, that's when rapid depolarization begins, because the voltage-gated calcium channels open and the positive is flowing into the cell until it reaches action potential. And then that's when the voltage-gated calcium channels close and the voltage-gated potassium channels open, and then the positive potassium that's inside the cell begins to flow out of the cell, and the cell becomes more negative until it gets back down to negative 58. So it's repolarizing back down to negative 58, and it'll be at resting potential again, and then the next impulse can start. So this picture of the heart is showcasing the specialized cardiac cells. So it would show that the SA node here is where an electrical impulse begins, and then it travels across the atrium to the AV node, and then it will go down the bundle of His until it reaches the Purkinje fibers, and then that's when it'll reach the actual cardiac muscles that contract. Cardiac muscle fiber action potentials. The cardiac muscle fiber, or myocardial cells, action potential phases are an initial rapid depolarization, so the sodium channels open, and then a small initial repolarization where the voltage-gated potassium channels open, and then a plateau where the voltage-gated calcium channels and the potassium channels are both open at the same time, and then a repolarization where more potassium channels are open and then the calcium channels become inactive, and then it reaches resting potential. And a voltage-gated potassium channels open is depolarization, and then the calcium and potassium channels open. The membrane potential plateaus at 15 millivolts for 200 to 300 milliseconds due to a balance between the slow influx of calcium and efflux of potassium. And more potassium are open, and then the repolarization occurs the rest of the way. And so on this graph here, you can see that the initial rapid depolarization is caused by the sodium channels opening and sodium flowing into the cell. And then right here is an initial rapid repolarization where potassium flows in, but then some calcium channels open at the same time that the potassium channels are open, and the potassium is flowing out while the calcium is flowing in, and they basically balance out, so no repolarization is happening. And then at this point the calcium channels close again, and only the potassium channels are left open, and so then it rapidly depolarizes the rest of the way back to resting potential here at 90. And then this graph is showing the same thing. Right here is the rapid depolarization, and then a short little repolarization caused by potassium, and then this plateau here where the calcium and potassium channels are both open. And then right down here is the rapid repolarization to resting membrane potential, because only the potassium channels are open, and potassium is positive and is flowing out of the cell, so the inside of the cell becomes more negative. And cardiac muscle fiber action potentials are about 100 times longer in duration than the action potentials in regular skeletal muscle fibers, and the long phase, or the plateau, of the action potential is caused by voltage-gated calcium channels. The action potential lasts about as long as the physical muscle contraction, and the cardiac muscle cells have a resting potential of negative 90 millivolts, and they are depolarized to threshold by action potentials from the SA node. And so this is the same graph that we saw on the other page, with the rapid depolarization because of the sodium channels, and then the initial, the plateau, and then the rest of the repolarization because of the calcium and the potassium channels. And this graph down here is an overlap of the electrical signal and the physical muscle contraction. And so you can see that the rapid depolarization coincides with the physical contraction of the muscle. And then as the muscle begins to relax, that is repolarization of the cardiac muscle cells. And the plateau here, the point of that is because when the muscle contracts, the blood is pumped out, and then when it relaxes is when the blood is flowing into the ventricle. And so the ventricle relaxation has to be longer to give the blood time to flow in, which is why it's about 100 times longer than regular skeletal muscle. And that plateau, or the calcium gates that cause the plateau, are responsible for that little break that gives the blood time to flow in. Electrolytes, electric activities, and body fluids. Electrolytes in human body fluids. Ion concentrations are measured in millimoles, and total osmolarity of extracellular fluid and intracellular fluid is about 300. So up here, this chart is the cations. Sodium is about 145 extracellular fluid and 12 intracellular fluid. Potassium is 5 extracellular fluid and 150 inside the cell. Calcium is 5 outside the cell and 10 to the negative 6th in the cell. And then down here you have the anions, and so Cl is 113 extracellular fluid and 4 intracellular fluid. HCO3 is 27 extracellular fluid and 8 in the cell. HPO4 is 2 outside the cell and 95 intracellular fluid. And then protein is 13 extracellular fluid and 55 intracellular fluid. And then these two at the bottom here are impermeable or fixed anions, including organic phosphates and other organic anions. And so the reason that the concentrations inside and outside the cell are important are because the membrane is permeable and these move on a concentration gradient. And so if it's higher outside the cell and the membrane is letting it through, then it'll move inside the cell automatically. These two down here at the bottom are fixed anions, which means they're too big to pass through the cell membrane, and that's important because they are anions, and so they are negative, and this is the reason that inside the cell usually stays positive when it's at resting potential. Osmosis. So as you can see, the molarity inside the cell is at 300, and so osmosis is when the solvent moves and not the solute, and so like the water would be moving in this picture here, and it's driven by the ion concentrations. And so if one side has a high ion concentration, then water, or whatever the solvent is, will move towards it to even it out. And so if it's hypotonic, then there is less than 300 in the cell, and so then water will move in. And then if it's hypertonic, there's more than 300 in the cell, and water will move out of the cell. Membranes and transport. Cell membrane separates the intracellular fluid from the extracellular fluid. The cell membrane is selectively permeable. Energy requirements for transport through the cell membrane. You have passive transport with a net movement down a concentration gradient, and active transport with a net movement against a concentration gradient, which requires energy. So the cell membrane is made up of a phospholipid bilayer, and so that's how it keeps the intracellular fluid from the extracellular fluid, and it's selectively permeable, which is how the ions can move between the extra and intracellular fluids. The energy requirements, passive transport does not use energy because it's following a concentration gradient, and so it'll just go from a high to a low concentration automatically. And then active transport uses energy because it has to move against a concentration gradient, because naturally the ions want to move from high to low, but if the cell needs it to move from low to high, then it has to use energy to do that. Passive transport. Diffusion occurs if there is a concentration difference across the membrane for the substance, and the membrane is permeable to the diffusing substance. If a substance is allowed to diffuse to equilibrium, the molecules will be distributed uniformly on each side of the membrane. So diffusion, unlike osmosis, is where the solute is moving, and so if there's a high concentration of the solute, then the solute itself will move to a lower concentration, and that's what this is saying. If there's a concentration difference, that's what drives the movement. The membrane is permeable, so it doesn't use energy to move back and forth. And then this one is saying that if there is a concentration difference, then the solute will move down the gradient until it's equal, and then it'll stop moving, basically. Solute diffusion. On this side over here, you have a high concentration and a low concentration, and so the net diffusion, this arrow is thicker going this way because it's moving from a high to a low concentration, and it'll do that until it reaches over here where there is no net diffusion because the solute is equal on both sides. The ion concentration is the same on both sides, so there's no need for it to move back and forth. Although some solutes can diffuse through the cell membrane, charged ions must diffuse through pores in the cell membrane called channels. So here in this picture, this part in the middle is the cell membrane, so the phospholipid bilayer. And then you have the inside and the outside of the cell, and the ions here are moving through this big yellow thing, which is an integral protein that is acting as a channel for the charged ions to move through. And so they will still be moving down a concentration gradient from high to low concentration, whichever is outside or inside the cell. Whichever one is lower, that's the direction they'll be moving. An example of this kind of channel would be the voltage gadolinium calcium channels that the muscles use for depolarization. The rate of diffusion. Diffusion rate depends upon the magnitude of the concentration gradient, the relative permeability of the membrane to different ions. Neuronal cell membrane is 20 times more permeable to potassium than to sodium. Temperature, a higher temperature means a faster diffusion rate, and then surface area. So the magnitude of the concentration gradient, if there is a bigger difference in the concentration inside and outside the cell, then the ions will move more quickly down the gradient, as opposed to if it was just like a few ions off. Relative permeability of the membrane to different ions, and then temperature. If there's a higher temperature, there's a faster diffusion rate, because when the temperature increases, the molecules are more active, and then therefore they're more likely to move across the membrane down the gradient. And then a surface area. If there's a higher surface area, if there's more space for the ions to move in and out, then they'll move out more quickly and more often. Active transport. Movement of molecules or ions against their concentration gradients from lower to higher concentrations It requires ATP, and there are two types of active transport, primary and secondary. While passive transport could be compared to gravity, like it just falls down and it's easy, it doesn't require any energy, active transport could be compared to like an elevator or something that's working against the natural force, like of gravity, or against the concentration gradient, and therefore it has to work harder and use energy to move whatever it needs to move. Primary active transport is the movement of ions or molecules, for example a sodium ion or glucose, through a cell membrane, requiring the direct expenditure of energy, in which ATP hydrolysis occurs at the same site as the movement of the ions or molecules through the membrane. So in this little animation here, you can see that in order to move the ion in or out of the cell, an ATP is used in combination with a high energy phosphate to turn ATP into ADP and move the molecule through. So primary active transport, this is another example of it. ATP is required for the function of the carriers. The molecular ion binds to the carrier site, and then the binding stimulates the phosphorylation or the breakdown of ATP. Conformational change moves the molecule to the other side of the membrane. And so here you can just see a picture of it. An example of where this would be used is during the depolarization and repolarization of the SA node, because calcium is higher outside the cell, and so to depolarize it and make the inside of the cell more positive, it floods in. And that is used, they use a concentration gradient for that, but then before the next depolarization can happen, somehow the calcium has to get back outside of the cell, and that's where the primary active transport comes in, and they have to use energy because they're going against the concentration gradient this time. The sodium-potassium ATP ACE pump is the second example that we have of primary active transport. The carrier protein is an ATP enzyme that converts ATP to ADP in 1-phosphate. 3 sodium ions are pumped out for every 2 potassium ions pumped into the cell, and the pump is electrogenic. So in this picture you can see the potassium being pumped into the cell and the sodium being pumped out, and this would be used in the myocardial cells, because when the myocardial cell depolarizes, the sodium is pumped into the cell, and then during repolarization the potassium is pumped out, and so those are used on a concentration gradient. So then to get them back to where they need to be, the potassium has to be pumped back into the cell and the sodium has to be pumped out of the cell using this form of active transport, so that it can get its concentrations back to where they need to be for the next action potential to be received. Secondary active transport mechanism, to the right is the sodium-potassium ATPase pump, which transports 3 sodium ions out of the cell and 2 potassium ions into the cell with each ATP hydrolysis. This creates a concentration gradient for sodium, which has a higher concentration outside the cell, which drives the secondary transport of a glucose, an example of a supporter of the cell, in conjunction with the movement of sodium ions along its concentration gradient. And so in this picture here, this would be the sodium-potassium ATPase pump that we saw in the last slide, and you can see that ATP and the phosphate are being turned into ADP in order to move the ions outside of the cell here. And then these little shapes here are glucose, and they are binding with the ions and bringing them back into the cytoplasm there. Membrane potential. Proteins and phosphates are negatively charged and cannot cross the membrane. These anions attract positively charged cations that can diffuse through the membrane pores. The effect of fixed anions on the distribution of cations. So this box here is representing the fixed anions, and those are the proteins and stuff that are too big to move through the cell membrane, so they're stuck inside the cell, and since they're negative, they have an electrical attraction that pull the cations, or the positive ions, in towards the cell. And then once the positive ions build up in the cell, then a concentration gradient allows them to go back out of the cell. Potassium equilibrium. So here on this, you can see there's an electrode inside the cell, an electrode outside the cell that's reading the volts, and it's reading -90 because that's where equilibrium is. So here again you have the fixed anions, and they're pulling in this cation, potassium, in towards them, and here it's building up and there is more potassium inside the cell than outside, so then it's using diffusion to go outside. Potential difference of -90 millivolts if potassium were passively distributed across the membrane. So that means once it's all evenly distributed, then it should come to resting potential at -90 millivolts. Resting membrane potential. High permeability to potassium. Resting membrane potential is slightly less than equilibrium because some sodium can diffuse into the cell. Membrane is slightly permeable to sodium. The sodium-potassium ATPase pump is electrogenic. So this last bullet is saying the sodium-potassium ATPase pump is electrogenic, so that means it uses this, like this kind of picture that we just saw, to maintain the resting potential, and so the sodium is attracted in by the fixed anions, and then it uses the concentration gradient to go back out, and this is just used to maintain the equilibrium of sodium and potassium. Resting membrane potential. This shows a picture where sodium and potassium are both going in and out, and it has the fixed anions on there, which the pump is electrogenic, and so that's what that means. The fixed anions are causing it to go in, and then the concentration gradient causes it to go out. And here you can see the voltmeter is -65 millivolts because that's where it's going to be at resting potential. Circulatory system, cardiac cycle, and EKG. The cardiac cycle, so systole is contraction, diastole is relaxation, and atrial systole is during the last 0.1 seconds of ventricular diastole. And so you can see in this picture here, the ventricles are contracting while the atria is relaxing, and then the ventricles are relaxing and filling up with blood while the atria is contracting. And the ventricular systole is only 0.3 seconds, but diastole is 0.5 seconds because it has to be longer in order for the blood to be able to go in and increase in diastolic volume. Isovolumetric is relating to or being an early phase of ventricular systole. The cardiac muscle exerts increasing pressure on the contents of the ventricle without significant change in the muscle fiber length, and the ventricular volume remains constant. So this is basically just saying that even though the pressure is increasing, the actual length of the muscle is not changing. It's not getting bigger or smaller. The heart sounds. The first heart sound is made when the ventricles contract, and that's the sound of the two atrioventricular pumps closing at the same time. And then the second heart sound is made during ventricle relaxation, and that is the sound of the semilunar valves closing. Ventricular pressure changes during cardiac cycle. 1. Isovolumetric contraction and first heart sound, which is the AV valves closing. Then number 2 is ejection phase. 3. Ventricular pressure falls and the second heart sound, which is the semilunar valves close. Isovolumetric relaxation lasts until pressure in ventricles falls below the pressure in the atria. 5. Rapid filling phase of the ventricles. And 6. Atrial systole ejects final volume of blood into the ventricles. And so these two graphs here, the first one is showing the pressure, and then the second one is showing the volume. So the first heart sound is made when the AV valves close, and you can see that this is when the pressure is rising. And then 2 here is the ejection phase, so blood is being ejected from the ventricles, and that's when the pressure is the highest. Number 3 here is when the ventricle pressure begins to fall, and it's also when the second heart sound comes in, which is the semilunar valves closing. And then 4 and 5 here is when the ventricles are relaxed. You can see that the pressure is really low, and 5 represents when the ventricle is completely relaxed and the blood is flowing into the ventricle, so rapid filling phase. And then number 6 is when the atria contracts and gets the rest of the blood down into the ventricle. Electrocardiogram, EKG, ECG. EKG records potential differences across the surface of the heart. P, QRS, and T waves. ST segment and the action potential. So on this graph here you can see the EKG, and this is an indirect measurement of the electricity that is going through when the cell, the heart muscle, is depolarizing. So here the P is atrial depolarization, so the atrium's contracting and sending blood down into the ventricle. And then the QRS here, this spike, is representing ventricle contraction, and then T is ventricle repolarization. So QRS is ventricle depolarization, and then over here T is ventricle repolarization. In EKG, the potential difference across the atria creates the P wave. When about half of the atrial muscle fibers are depolarized, the P wave reaches its peak amplitude. So you can see that in this picture because it starts to go across the atrium and the P wave is beginning, and then when it's about halfway filling the atrium, you can see it spikes a little bit right there, and then it's going back down as it's filling up the rest of the way, as it's moving the rest of the way across the atrium. EKG. The delay between the P wave and the QRS complex is the time required to stimulate the AV node and eventually the bundle of His and the Purkinje fibers. Depolarization of the ventricles occurs from the inside toward the outside and produces the QRS complex. Repolarization occurs from the outside toward the inside and produces the T wave. So you can see here that the delay between the P wave and the QRS complex is when the signal is traveling down to the bundle of His and the Purkinje fibers. So you can see that the atria has already been stimulated, and then it's traveling down the ventricles here. And so then you have the QRS as it goes down the ventricles, and then the depolarization of the ventricles comes from the inside towards the out and produces the QRS complex, so the spike on the EKG there. And then repolarization occurs from the outside in and produces the T wave over here. EKG and heart sounds. The first heart sound is produced just after the QRS complex, and the second heart sound is produced shortly after the peak of the T wave. So these two graphs here are basically just illustrating that. The top one is showing the pressure in the ventricle, so that would be like the physical contraction of the muscle. And then the bottom one here is showing the EKG, which represents the electrical side of the contraction, so the repolarization and depolarization and that kind of stuff. So it's relating it to the heart sounds, and so the first heart sound is just after the QRS complex, and so that's when the ventricle is contracting, because the first heart sound is made when the AV valves are shutting. And then the second heart sound is right in the T wave here, and that is when the ventricle is relaxing. It's during the repolarization of the ventricle, because that is the sound of the semilunar valves closing.